Good morning, everyone. As a continuation of our immunology series, we are seeing the most important cell, that is the T cell. So, what is a T cell? T cell is a part of the innate immunity. T cell is a naive T cell. This is a TH0 or this is a naive T cell. What does a naive T cell mean? It means it hasn't differentiated into either CD4 or CD8. So if it has, the naive T cells can get differentiated into CD8 and the CD4. This CD4 will again get differentiated into four types of CD cells. It can get differentiated into helper T1 or helper T2 cells or helper 17 or Treg cells. So that CD4 which hasn't differentiated into either of these are known as naive T cells. The CD8 is also called as a cytotoxic T cells. CD4 is called as helper T cells. So why should it be called as cytotoxic? Why should it be called as helper T cells? It is called as cytotoxic because once it recognizes the MHC class 1 molecule which is present on normal cells, it is called as a cytotoxic T cells because once it comes in contact with the MHC class 1 which is present on the all the nucleated cells, then it will attack the cell and then it will kill the organism or the pathogen that is present inside. This is the role of CD8 or a cytotoxic T cells. Since it attacks immediately, it is known as cytotoxic. But whereas CD4 or the helper T cells, once it comes in contact with the MHC class 2, which is expressed on the antigen presenting cells, then it will help the antigen presenting cells, be it a macrophage or else a B cell, to destroy the pathogen that it has phagocytosed by itself. This is the reason why CD8 is known as a cytotoxic, but CD4 is known as a helper T cells. Now we are going to talk about the two cell activation model. How does the cytotoxic as well as the CD4 cells get activated? So if you see here, this is the MHC class 1 and this is the beta 2 microglobulin which stabilizes the MHC class 1. MHC class 1 will combine with CD8 T cells. The CD8 T cell will have a T cell receptor as well as this is the CD8 which is present on the T cells. So what does the CD8 mean? The CD8 is present on the T cells and this is the T cell receptor and it recognizes the MHC class 1 which is carrying the peptide. So this is the primary signal. What is the signal for the T cell receptor? That is finding the MHC class 1 carrying the abnormal peptide or the pathogen particle. Similarly, here we have the MHC class 2 and this is the T cell receptor and this is CD4. Fine. So the CD4 will be something similar to this. This is a T cell receptor and here we have the cell, that is the T cell. So what is the primary signal for CD4? Again, this MHC class 2 and the abnormal peptide that it is carrying is the primary signal. Following this, there will be a co-stimulation or there will be a secondary signal. So what does this mean? The antigen presenting cells will be presenting as well as the nucleated cells. All the cells will be having a CD80 or CD86. It CD80 is called as B71 and the CD86 is called as B72. They are present on antigen presenting cells. They will bind to CD28 which is present on the T cells, providing the second signal or the confirmation for full activation. What do you mean by this? By this is once this primary signal has activated, the T cells are ready to destroy the cell or to mount the immune response. But the immune response it mounts has to be legitimate. It cannot kill a normal cell. For that, it requires a secondary signal that is given by the CD80 or 86, which the signal present on the antigen presenting cells. But CD28 is the signal which is present on the T cells. So when this secondary signal has taken place, then we have, see, this is the CD28 which is present on T cells and this is the 8086 which is present on antigen presenting cells and also on the nucleated cells. So this is necessary for, this is a co-stimulation or the secondary signal for further activation of the T cells. Having said that, This is the T cell and this is the recipient antigen presenting. Here we have the MHC complex, that is a primary signal, and again we have the co-stimulation, that is the secondary signal, which happens by CD28 on the T cells and CD80-86 on the antigen presenting cells. So with this, the secondary stimulation happens, and now the T cells have the full confirmatory message that you are ready to launch, you are ready to burst, you are ready to whatever, activate the antigen presenting cell, whatever it is, you are ready to proceed and take the next step of killing or activating or mounting the immune response. So this is how the T cells are stimulated. What is the consequence of T cell activation? Once the CD8 is being activated, that is, once the CD8 is activated, that is, the cytotoxic T cells are activated, that will directly target and eliminate the nucleated cells that are displaying the foreign antigens on MHC class 1 molecules through apoptotic mechanisms. And once the CD4 T cells are activated, the CD4 T cells will undergo differentiation, enabling diverse effector function based on the specific helper T subtype that it emerges. So here we are going to see about the CD4 and the different T cell differentiation. So the T helper 1 cells, so this will activate the macrophages. So the T cells, the CD4 T cells can now differentiate into 4 main types of T cells. That is helper T1, helper T2, as well as T helper 17 and the Treg cells. What is the helper T1 or the T helper 1 does? It will activate the macrophages and it will eliminate intracellular pathogens. Just learn about it in brief now. I will tell you in detail in the subsequent slides. But the T helper T2 CD4 cells will stimulate the beta cells to produce, the B cells to differentiate into plasma cells, thereby producing antibodies. While the Treg cells will suppress immune responses to prevent autoimmunity and maintain immune tolerance. The 17 T helper 17 cells will promote neutrophil production and recruitment, which are essential for inflammatory responses. Let's see how the helper T cell naive CD4 is converted to is forming the T helper 1 cell. So this is the naive T cell that has come in contact with the macrophage. The macrophage has phagocytosed the pathogen, it has processed the peptide, and via its MHC complex, it is presenting that peptide to the T cell receptor. This T cell, that is the CD4 T cell receptor, is also getting stimulated with this MHC complex 2 presentation. What happens next? The macrophage will not only express the MHC2, but it will also be producing a cytokine environment that is rich in interleukin 12. With this interleukin 12, it will also activate the T cells to get converted to T helper 1 cells. Once it gets converted to T helper 1, it will be producing interferon gamma. This interferon gamma will convert the macrophages into an activated macrophage such that it will be producing more interleukin 2 and more T helper 1 cells are formed, which will be forming more interferon gamma, and therefore this is a continuous powerful positive feedback loop. This activated macrophages will have an enhanced pathogen killing and it will kill the pathogens that it has phagocytosed by itself. This positive feedback loop is crucial for USMLE step 1. This is what is about T helper 1 cell development. In the previous slide, we understood that interferon 12 as well as interleukin 12 and interferon gamma will stimulate the macrophage activation and enhance the microbicidal activity. This persistent antigenic stimulation will lead to sustained inflammatory responses. Now, the macrophages, because of sustained inflammatory response, the macrophages are all fully activated. They will organize into dense clusters called as granulomas with cells which are termed as histiocytes or epithelioid macrophages. So with this, we have seen about how the T helper 1 cell is formed, and that is by the activation from the macrophages. Next, we will see how the T helper 2 cells are formed. The T helper 2 cells are formed by when it comes in contact with the beta cells. So the interleukin 4 and 10. Previously we saw interleukin 12 as well as interferon gamma are necessary for T helper 1 cell formation. But here interleukin 4 and 10 will drive the CD4, naive CD4 into T helper 2 cell differentiation. This is helpful for humoral immunity. How? See, this is the B cell. This is an antigen presenting cell and it has a MHC class 2. It has the phagocytosed peptide which is loaded on the MHC class 2 and presented to the T cell receptor. This T cell receptor is the naive CD4 which has a CD4 receptor over here. I mean the CD4 over here as well as the T cell over here. This is the primary stimulation. The secondary stimulation is, as we had seen previously, by the CD8 which is present on the T cells as well as CD80/86 which is present on the antigen presenting cells. After this, the T cells are all ready. In this environment where there is interleukin 4 and interleukin 10, it will form the differentiation of this naive T cell into T helper 2 cells. In addition to this, for the B cells, there is an extra ligand activation for B cell to be activated. What is that? There is a CD40 ligand and a CD40 receptor. The CD40 ligand is present on the T helper cells. That is attached to the receptor, which is CD40 receptor, which are present on the B cells, thereby providing essential co-stimulatory signals. Once the CD40 receptor and the ligand are attached with each other, it will result in antibody formation by the B cells. The activated B cells will differentiate into antibody secreting plasma cells, thereby targeting extracellular pathogens. In addition to this, the antibodies or the B cells will undergo somatic hypermutation as well as affinity maturation, which I have explained in the previous slides, previous PPT, and I have added it in the playlist. To ensure the helper T1 and the helper T2 balance, for the helper T1 we have interleukin 12 as well as interferon gamma. This is interleukin 12. So how do you remember interleukin 12 and interferon gamma? There is a key, there is a mnemonic like 1, 2 gamma helps interleukin 1, while 4 and 10 deteriorates, suppresses interleukin 1, and the reverse is true for T helper 2 cells. 4 and 10 helps T helper 2 cells, 1, 2 gamma suppresses T helper 2 cells. Here it is interleukin 12 and not interleukin 2, and this is interferon gamma, which helps in the formation of T helper 1 cells but suppresses T helper 2 cells, because we don't want both at the same time, right? Similarly, interleukin 4 and interleukin 10 will help T helper 2 cells, but it will suppress the T helper 1 cells. Whereas CD40 ligand and the 40 interaction is particular for plasma B cells.